Dear students, today we will talk about enzymes. The study of enzyme is called enzymology. Enzymes are specialized class of protein which are responsible for catalysis of chemical reaction within living body. Enzymes are called biocatalyst. Now let's understand what is the role of catalyst. The catalysts are the chemicals that usually enhance the rate of chemical reaction. They are not consumed during the reaction. So this is how they behave. They are one of the reactant. They participate in the chemical reaction, but they are not consumed. Similarly, is with the case of enzyme. Similarly, enzyme also enhance the rate of biochemical reaction, but the enzymes are highly specific in catalyzing the reaction. So this is how they differs from catalyst. Now how does a chemical reaction differs from a biochemical reaction? Let's talk about this. In chemical reaction, the reactant react under a specific condition and final end product is formed. The reactant and product are compound or molecule. During biochemical reaction, the initial reactants undergo transformations to form end product. The reactant and product are complex organic molecules. Sometimes the series of reactions are required to convert the initial reactant to the final end product. So this is just to give you an idea that how the chemical reaction differs from the biochemical reaction. Now the reactants that are used or that are present in the biochemical reactions, they are called substrate. The human body is a complex system of biochemical reactions. If these reactions occur in absence of enzyme, they would be too slow to sustain living body. Hence, life cannot exist without enzymes. Okay, so they enhance the rate of reaction. Hence, the reaction proceed uh, at faster rate, and hence there are so many metabolism which are going at this rate, and they sustain the living body. Now, can enzyme catalyze the reactants outside the body? Let's talk about this. The enzymes are usually produced by the cell as per the requirement. So whenever there is requirement, the cell will produce the enzyme, but they can retain their catalytic activity or action even outside the cell or in the cell extract. Therefore, the enzymes are used in vitro and in vivo studies of chemical reactions, or you can say biochemical reactions. So, if you isolate the enzyme from the living body, and if you catalyze, if you do the reaction in the test tube with the desired substrate, then it is going to convert the substrate to the product in the test tube. So this we call in vitro study, since it is done in the test tube. Now let's briefly focus on the discoveries. The oldest known enzyme reaction is the alcoholic fermentation. Louis Pasteur identified that sugar fermentation is caused by living a yeast cell, Persos and Payen. Okay. Now they discovered the enzyme dietase, which was known to be the organic catalyst. The next is Willem Kuhn. He coined the term enzyme. Edward Buckner demonstrated that a dead yeast extract could carry out the same fermentation reaction as in the living cell. So he carried out the fermentation without yeast, living yeast, but with the extract which is prepared from the yeast. So that contains the enzyme, and it carried out the fermentation reaction. Now let's discuss the structure and composition of enzyme. The enzymes are basically globular protein of 60 to 2500 amino acid. Now there is ribozyme, which is RNA based enzyme. Usually all the enzymes are protein in nature. So they have 3D configuration, 3D structure. Just we have discussed the 2D, 1D and 3D structure of protein, that is primary, secondary and tertiary. So they have 3D structure of protein. During biochemical reaction, the reaction occur at specific site of an enzyme. Now instead of the whole enzyme, a part of enzyme participate in the reaction. This is the site. So this, we are talking about a particular site where the substrate bind covalently, and this site is called active site of the enzyme. Active site is also associated with specific set of amino acid. So it is different for different enzyme and had 3D configuration. So basically if you see the structure of enzyme, it is 3D folding of the protein, and a part on this protein act as an active site, which again has a some 3D configuration to bind with the substrate. For example, glycine, histidine, alanine. Okay, now these are the specific amino acids which are present at the active site of the enzyme aldolase. Now, if you look at the structural classification, they are divided into two categories, simple and conjugated. The simple enzymes are only formed of the amino acids. They only contain amino acid. For example, pepsin and trypsin. Okay. Then we have conjugated, which has two different parts. One is protein part, another one is non-protein part. The protein part is called apoenzyme, and the non-protein is called prosthetic group. Protein part is usually larger part of the enzyme, and non-protein part again is of different type. They may contain cofactor or coenzyme. So this non-protein part is also a essential part of the enzyme, because here they consist of either cofactor or coenzyme. Okay. So what are cofactors? They are inorganic metal ions such as magnesium, zinc. So there are certain enzymes which will only work when you supply, or when they have magnesium or zinc or manganese ions. Okay. Cofactor and coenzyme. Coenzymes are the organic ions synthesized from the vitamin B complex and vitamin C. So certain enzymes have requirement for coenzymes such as FAD, NAD, FAD, FMN, NADP, coenzyme A, thymine. Okay, so all these are synthesized from the vitamin. So coenzymes synthesized from the vitamin B complex, and cofactors are mineral ions. So these are the prosthetic part of the enzyme. Okay, later on we will see how they participate in the reaction. Now if you look at the occurrence of the enzyme, enzymes occur in many different ways. They may occur inside the cell called endoenzyme, they occur outside the cell called exoenzyme, and usually the enzymes are produced by certain cells and then they are secreted. So they may be secreted inside. Secretion means they are produced and released, that is called secretion. So they may be secreted inside or outside. Inside, for example, the enzyme those break down the glucose. Okay, so all those enzymes are present in the cytoplasm inside the cell. Whereas if you see hydrolytic enzyme which participate in digestion, they are all secreted by certain cells, and their content is poured in the lumen. Okay. Similarly, most of the microorganism, they secrete the enzymes outside in the medium in which they are growing. Okay. Just even if you see yeast, it secrete the enzyme outside in the medium. That's why yeast extract could be used as the factor which can cause the fermentation. Then we have the occurrence in the inactive form. So usually some of the enzymes they present they or they are secreted in the inactive state. For example, the hydrolytic enzyme, okay, they are secreted in the inactive state, because if they are secreted in the active state, they will digest all cell content. Okay, for example, pepsinogen. So after some time when the protein come, it become active and forms the pepsin. Then isoenzymes are there. Now some enzyme occur in different molecular structure but have same catalytic function. So their structure differs but their function is same. For example, lactate dehydrogenase, it is present in different part of the body, okay, but catalyzes the same reaction. Then we have allosteric enzymes, and these are called regulatory group of enzymes. Then let's discuss the nomenclature. So if you see the nomenclature and classification of the enzyme, so basically the enzymes are classified, or you can say they were named initially on the basis of the type of substrate, type of chemical reaction, or type of product. Okay, so the enzymes are usually named by adding the suffix ase to the substrate on which they act. Okay, so this is how they are named. For example, the enzyme which act on the lipid are called lipase. Then second is type of reaction. For example, an enzyme is catalyzing the reaction oxidation. Okay, so the name is oxidase. Okay, and the enzymes are also classified on the basis of product form. For example, rhodonase, it catalyzes the formation of rhodonate from hydrocyanic acid and sodium thiosulfate. Then later on, International Union of Biochemistry has classified the enzyme into major 6 categories and given a specific number to a specific enzyme so that we can identify them. So we will be discussing here the 6 major categories: oxidoreductase, transferase, hydrolase, and then we have lyase, isomerase, and ligase. So we'll be discussing this. First is the oxidoreductase. Now all the enzymes which fall into this group, okay, what kind of reaction they catalyze? They catalyzes oxidation and reduction reaction by removal of hydrogen from the substrate or metabolite. You know, the oxidation is what? Addition of oxygen is called oxidation, removal of hydrogen is called oxidation, and loss of electron is called oxidation. So any enzyme which is doing all these things, oxidation and reduction, in any way, they fall into the category oxidoreductase. So just to explain you, X1 and X2 are the substrate, and the enzyme, what it is doing, it is either removing the electron, or it is adding the oxygen, or it is adding the hydrogen. So if you look at the substrate A, which is in the oxidized state, okay, so hydrogen is removed from that by the enzyme, and now the X1, if you see in the product, is converted into the reduced form. So the product is the reduced form of X1 over left hand side, over here it is substrate, whereas at right hand side it is a product. Okay, so for example, cytochrome oxidase, so is the enzyme which catalyzes the reaction. Cytochrome C is a substrate, and this enzyme usually what it do, it add oxygen to cytochrome C and catalyzes the reaction oxidation. So the enzyme is known as cytochrome oxidase. Okay, next enzyme is transferase, which catalyzes transfer of specific functional group other than hydrogen. Okay, so it is not doing hydrogen and oxygen transfer, but other than that, certain transfer of group. So say substrate X1 is attached to certain group. Now this group is transferred to X2. So the product is, the X2 is now having the group. Okay, so this is done by the enzyme, and this enzyme comes under the category of transferase. For example, hexokinase is the enzyme. Now what it is doing in this reaction, the hexokinase act on the glucose, and what it is doing in presence of ATP, so whatever phosphate is there from the ATP is going to transfer over the glucose, and now the glucose is converted to glucose 6 phosphate. Now hydrolase, it catalyzes the breakdown of large complex molecule into the smaller one by adding the water molecules. So this reaction, these kind of reaction we have seen several time in digestion, we have seen in biomolecules. So let's say X1, X2, they this is one particular you can say compound and acted upon by the enzyme in presence of water. So water is going to add between two, one side OH, one side H, and two different product are formed. For example, sucrase and sucrase act on sucrose and it gives glucose and fructose. Now another enzyme is called lyase. It catalyzes breakdown of compound and removal of group from the compound without hydrolysis. So there is no hydrolysis. A group is attached to X2 in presence of enzyme. This is removed and a bond is created. Uh, Bond is broken down and uh, they are separated. So for example, fructose 1,6 biphosphate, okay, so it get broken down into two different compound, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate in presence of enzyme aldolase. Isomerase, it catalyzes intramolecular arrangement of atoms in molecule. So isomerism takes place in presence of enzyme. X1 is a substrate, so it will have different structure. Any kind of isomerism which is catalyzed by the enzyme, they all fall into the category isomerases. So glucose 6 phosphate is converted to fructose 6 phosphate in presence of enzyme phosphofructo isomerase. Then ligase, it catalyzes joining of two different molecules. So X1 and X2 are two different substrate. They are joined either by phosphate bond, nitrogen bond, oxygen, carbon or sulfate bond. For example, the enzyme pyruvate carboxylase. So what it is doing? It is creating the bond between the pyruvate and carbon dioxide and it forms another compound which is called oxaloacetate. Okay, so these are different categories of enzymes and they catalyzes different type of reaction. So they are classified into 6 major groups. So next we will continue in the next video. Thank you so much.